In this presentation, we are going to examine genome and proteome. A genome is a set of all proteins that are coded for. Human genome has about 3 billion bases and about 25,000 genes. A genome is considered to be static, but a retrovirus may add genes to the genome. A proteome is a set of proteins that are expressed under given conditions. It is a subset of genome and it is not fixed. And because proteome is subset of genome, at first glance one may think that proteome would be smaller than genome because only a part of genome is expressed at a time. But actually proteome is larger than the genome because proteins can be chemically modified in post-translation modification, and also proteins can aggregate into larger units. An example of activation and deactivation of a gene is the gene that codes for fur color in cats. It is located on the X chromosome. Since males have XY chromosome, they have only one copy of the X chromosome, and then that gene may be expressed, which means that fur is going to be colored, or if it's not expressed, then fur is not colored and it's white. That means that male cats either have single color, they are solid colored, or have two colors, white and something else, like tuxedo cats could be black and white, or orange and white is another common combination. Cats that are solid orange are mostly male. In fact, I believe 95 or 98% of them are male, but they could also be female. So while cats of single color or two colors, where one is white, so black and white, orange and white, could be male or female. Lots of them are male, but they could be male or female. Cats that have three colors, like calico cats, must be female. That's because females have two copies of X chromosome, and only one of them, of course, is activated at a time, but at parts of the fur, one X chromosome may be activated, and other parts other may be activated. So cats that are, for example, black and orange must be female, because that means that two different X chromosomes are being activated on different parts of the fur. And of course, third color would be white, where neither of the chromosomes is activated. So, as I said, three-colored cats must be female because they must have two copies of the X chromosome. In this photograph, there is a female kitten, and she is actually exhibiting initially very early in her life, at a relatively young age of about 2 months old. She is exhibiting actually some sort of Siamese coloration. So her body is exhibiting brown coloration, and actually this is light, so this is she has been hit by the sun, so the fur color is a little bit lighter because of the reflection of the sun. But these are actually her more accurate colors. Nonetheless, you can see that her body is exhibiting brown coloration, clearly brown coloration, while face and limbs are exhibiting dark black coloration. That's because gene for black has been deactivated by the higher body temperature. And that's actually what is behind Siamese cat coloration. That higher body temperature deactivates gene that codes for black fur. Where body temperature is lower, such as face and extremities, that gene is active and fur has black coloration. As this kitten grew, actually one gene that coded for brown got deactivated and one that codes for black got activated. So at the age of about 4 months, which is this photograph on the far right, one can see that she has actually strapped coloration where brown is changing into black. And then as we follow her further, we can see that at the age of about 6 months, she is turning almost completely black. And also note that how interesting is that her fur in parts is turning white. So as gene that is coding for brown was deactivated and one that codes for black is not yet activated, that part of the fur was white. So here are two photographs of her at about 6 months. And finally, at 1 year, she turned completely black. As another example of gene activation and deactivation, this kitten actually suffered from asthma early in her life. And that's what these photographs show. It may appear to be cute that she is sort of panting with her tongue out, but actually that's asthma. That could be pretty dangerous. But her symptoms disappeared at the age of about 6 months. And so causes of asthma are not known, but they may be genetic. And again, that could be another example of change of gene expression, and that's why she was exhibiting asthma symptoms early in her life, but at the age of about 6 months, she actually turned into a completely healthy cat. Three-letter sequences or codons of DNA and RNA, here RNA is shown, encode for individual amino acids. So that's the genetic code. And so here genetic codes shows codons, those three-letter sequences that code for each of the 20 common amino acids. And also there are two additional codons, start and stop codons. Now, if we exclude amino acids that are altered through post-translational modification, there are two other amino acids that occasionally appear in proteins and that are coded for. One of them is selenocysteine, which is found in archaea, eubacteria, and animals. And it originates from modification of serine that is attached to transport RNA. The other one is pyrrolysine, which is found in archaea, and is most likely derived from lysine attached to transport RNA. Modification occurs after transport RNA is charged with appropriate amino acid, serine in the case of selenocysteine and lysine in case of pyrrolysine, and then both are coded for by codons that usually serve as stop codons. So selenocysteine transport RNA recognizes UGA codon, while pyrrolysine transport RNA recognizes UAG codon. Now, only a small fraction of UAG and UGA codons in messenger RNA are recognized as coding for selenocysteine and pyrrolysine, and what determines that is not clear. From the previous slide, genetic code, you have seen that for a number of amino acids, in fact for most of the amino acids, there is more than one codon that codes for that amino acid. And that ensures that genetic code is actually pretty robust, pretty stable, so that minor random mutations that occasionally may occur in DNA don't result in catastrophic mutation that will actually render protein that is synthesized inactive. And so most of those random minor mutations end up being silent mutations that don't affect function of the protein. That's because either as a result of mutation, same amino acid is incorporated, or very similar amino acid is incorporated. Very similar means that instead of one acidic amino acid, another acidic amino acid is incorporated. So instead of glutamate, for example, aspartate is incorporated. That wouldn't change function or properties of synthesized protein much. Or instead of one basic, another basic amino acid is incorporated, or instead of polar, another polar amino acid is incorporated, or non-polar is replaced by non-polar amino acid. Now, when mutation results in more serious, what you would call mistake, then we could end up with basically catastrophic mutation where protein becomes either non-functional or has very low functionality. One example is molecule of hemoglobin. And there are a number of mutations that are known in hemoglobin that are silent and cause no disease because they substitute one amino acid for similar amino acid. But here is an error that is quite well known error that replaces hydrophilic amino acid glutamate with hydrophobic one, and that's valine. And that's sickle cell disease. A single nucleotide mutation changes codon GAG to GUG. GAG codes for glutamate. And so there should be a glutamate amino acid residue that is on the surface of hemoglobin. But when that codon is changed to GUG, then that codes for non-polar valine. Valine is hydrophobic and it's not capable of forming hydrogen bonds with water. It has only hydrocarbon chain. And so resulting hydrophobic interactions, basically water tries to exclude valine from its environment. So resulting hydrophobic interactions force such mutated hemoglobin molecules to aggregate together. Basically, they aggregate together through interaction of those valine side chains on different hemoglobin molecules. And so they clump together and produce rigid fibers that deform red blood cells into sickle shape, and that's why disease is called sickle cell anemia. Okay, so this completes our presentation on genome and proteome.